Hello everyone. Today in the subject of medical microbiology, we are going to study about actinobacteria. So in this lecture, we are dealing with the topic of actinobacteria, where we will get a brief introduction about actinobacteria and outcomes will be you'll be able to understand what are the essential uh, qualities of actinobacteria and where it is employed. So actinobacteria are a phylum, mostly gram-positive bacteria. So you must understand that it is seen that the phylum that is mostly gram-negative is mostly harmful to humans, and rarely gram-positive bacteria are proven have been proven to be harmful or pathogenic to humans. Right. So it is a phylum of gram-positive bacteria. They can be terrestrial or aquatic. So they have the capacity to be terrestrial, to live on the land, or they also have the capacity to live in aquatic environments, in water also. Right. So they are of great economic importance to humans. But on the other hand, they are also economically very essentially important to humans, as because of agriculture and forests depend on their contribution to the soil system. So they contribute a lot in the forest by acting in the soil system. So each and every detail is essential when it comes to actinobacteria, and it plays a vital role for human beings, for the benefits of the human beings. Though human beings are working constantly on it and trying to explore it more. In soil, they help to decompose the organic matter of dead organisms so the molecules can take up a new by plant. So when any organism dies off, or when any organism is dead in the process, so they try to break down its organic molecules that are complex substances into simpler substances. So in this ongoing process, the complex molecules are broken down into simpler molecules. This is done so that the simpler molecules can readily be absorbed by the plants in general. While this role is also played by fungi, actinobacteria are much smaller and likely do not occupy the same ecological niche or ecological niche. So, role played by fungi is much larger than the role played by actinobacteria. So, first of all, they do not play a vital role in ecological niche, and you cannot see them widespread like fungi. It is found everywhere, and they are present on selected locations only. So this makes them a lot different from others. So, what they are trying to teach you here, Actinobacteria is a phylum of gram-positive bacteria. They can be terrestrial and aquatic in nature, and they are essentially very much important to human beings, as agriculture and forest depend a lot on their activity on the soil system, and it readily helps to decompose the complex organic matter into simpler substances, and they also play a vital role in ecological niche. In this role, the colonies often grow extensive mycelia, like a fungus would, and the name of an important order, phylum Actinomycetes, the Actinomycetes, reflect that they were long believed to be fungi. So, this bacteria has a special characteristic. And what is that special characteristic? It grows mycelium on a very extensive scale, and this is. But according to the microbiology, this is the characteristic of fungi. And fungi would behave in a very different manner, right? So, whatever the things are going on, whatever the scenarios are going on, so actinomycetes, they are for a very long period of time, they were believed to be fungi, and they were kept in their category, although they are not fungi, right? So, most of their functions resemble to fungi, and their behaviors also resemble to fungi. So, experts kept them in the order of fungi, although they are not fungi, right? So, some soil actinobacteria, such as Frankia, live symbiotically with plants whose roots pervade the soil, fixing nitrogen for the plants in exchange for access to some of the plant saccharides. So, they were found to be playing a very important symbiotic relationship also, right? And symbiotically, they are very much beneficial for the plants as their roots pervade the soil and assist in the nitrogen fixation that is essentially important for the plants. So, uh, in return to that, plants offer them shelter and provides saccharides that are readily present in the plant's body. So this whole scenario turns out to be really very different, and it was when it was discovered, it was a new phase of study uh, in the ecosystem or in the phase of actinobacteria, right? So other species such as many members of genus Mycobacterium are important pathogens. But here you can see that from the very early beginning, the actinobacteria is vitally playing all the beneficial factors and is a major benefactor for the human beings. So this scenario leads to a very different idea about the bacteria that are normally seen as harmful and pathogenic to the humans. So they do have some characteristics like fungi that are very much beneficial. Okay, they do grow mycelium. Okay, and they because of this speciality of also breaking down organic molecules, complex organic molecules into simpler substances, they were known to be fungi uh, for a very long period of time. And some soil bacteria like Frankia, they are also known to have evolved a very good symbiotic relationship with some plants, where they help in nitrogen fixation and plant saccharides they get in return for shelter. So, these are some of the important factors that are essentially very much important when it comes to actinobacteria. Actinobacteria produce a variety of secondary metabolites with high pharmacological and commercial interest. So, from the very early beginning, we have been studying that they do produce secondary metabolites with high pharmacological commercial interests. Okay, their interest is really very high, and the secondary metabolites and pharmacological molecules of high commercial interest they produce. So, from the very starting, you have seen that actinobacteria is playing a major role for the benefit of human beings. With the discovery of actinomycin, a number of antibiotics have been discovered from actinobacteria, especially from genus Streptomyces. So, why do we need new antibiotics? Because although the antibiotics have been a boom for our medical sciences, and medicine has been progressing since then, but the old medicines, the pathogens, when they are being used on those pathogens, they start to develop some sort of resistance. So, carrying out that scenario, we always need to have or we always need to secure some sort of new antibiotics that are very much new to the pathogens, because pathogens will not be able to transform themselves into resistant species at a very fast pace. So, discovered from antibiotics discovered from actinobacteria, especially from the genus of Streptomyces. They are widely distributed in soil with high sensitivity to acid and low pH. So, they can also be used as uh, soil acidity indicators, because they are highly acidic to acidic conditions. They are highly sensitive to acidic conditions and low pH. Actinobacteria have a number of important functions, right? Including degradation, decomposition of all sorts of organic substances such as cellulose. So, their main function includes is to break down organic molecules, complex organic molecules, into simpler substances, right? So, substances such as cellulose, polysaccharides, proteins, fats, organic acids, and so on. So, what they do, they break these organic molecules into simpler substances, which enables a plant to absorb them essentially in a very easy manner. Beyond the great interest, actinobacteria from their soil role, much is yet to be learned about them. So, scientists and microbiologists are constantly working on actinobacteria, although they have discovered a large about actinobacteria. A lot still remains to be discovered. Although currently understood primarily as soil bacteria, they might be more abundant in fresh waters. So, because they are primarily found in soil, but on the other hand, they are most largely found in fresh waters. So, scientists and experts are still working on their nature and discovering their new aspects and new dimensions, because they do have a lot of scope and we need to research them a lot. Actinobacteria is one of the dominant bacterial phyla and contains one of the largest bacterial genera, Streptomyces. So, it is found to be extremely dominant and widespread bacterial phyla and contains one of the largest bacterial genera known as Streptomyces. So, what is the main nature of Streptomyces? Streptomyces on the and other actinobacteria are major contributions or contributors to biological buffering of soil. They are also the source of many antibiotics. So they create a buffer in the soil which can resist the change in pH very easily, and they also contribute to the development of a lot of antibiotics. The Actinobacteria genus uh, Bifidobacterium is the most common bacterium in the microbiome of human infants. So, they are also uh, found in the microbiota or microbiome of human infants commonly. Although adults have fewer bifidobacteria, intestinal bifidobacteria help maintain the mucosal barrier and reduce lipopolysaccharide in the intestine. So, although in that quantity they are not very much present in adult human beings, but they are also present in a significant number. The bifidobacteria also maintain the mucosal barrier, uh, especially in the intestine, right? And reduce lipopolysaccharide in the intestine, because lipopolysaccharide or LPS is the main component of pathogenic bacteria in the cell wall. So, bifidobacteria uh, plays a major role in creating the mucosal barrier that is effective in nature. Although some of the largest and most complex bacterial cells belong to actinobacteria, the group of marine actinomarinales has been described as possessing the smallest free-living prokaryotic cell. Right. So actinobacteria, although is the largest group, but actinomarinales are also known to be smallest free-living prokaryotic cells. So they do have a lot of variation in their habit and habitat, and we have to work hard and learn more about their nature and about their different varieties. Some Siberian or Antarctic actinobacteria is said to be the oldest living organism on Earth. So Siberia and Antarctica are already the regions of the Earth that are not very widely explored because of their climatic conditions and the harsh scenario they have. Although scientists do reach there and do collect the sample sometimes, so what they have found that in such inhospitable climate and such tough conditions, no organisms can survive. But Actinobacteria was found readily present there, and they found it to be the oldest living organism on Earth. And how was that? Frozen in per- permafrost around half a million years ago. So when scientists, what they do when they reach Antarctica, they drill in the ice, okay, and they take samples, and and they take ice that is there since a very old period of time, millions and millions years ago. What was the type of atmosphere the Earth was having? Because when ice is frozen, the bubbles of air are captured inside it. So from that, they are trying to understand the climatic conditions and the environment that was present on Antarctica. But what they have found that in permafrost, and they found the actinobacteria frozen in them, right? And it was around half a million years ago. So they are known to be the oldest living organism on Earth, and they have been linked with the first living form of bacteria that were widespread on the Earth. The symptoms of life were detected by CO2 release from permafrost samples 640 kya or younger. So these samples, when they were releasing CO2, and it was studied and analyzed. General actinobacteria of medical or economic significance, are in class of Actinobacteria and belong to the order Actinomycetes. Right. So this is a very different aspect. They do have a lot of medical importance and that contribute towards their economic significance also. While many of these cause disease in humans, Streptomyces is notable as a source of antibiotic. Of those actinobacteria not in Actinomycetes, uh, Grand Gardnerella is one of the most researched. Classification of Gardnerella is controversial, and MESHA uh, is catalogs it as both gram-positive and gram-negative organism. So here, what they are trying to explain is that although the gram staining is widely used in the identification of microbiology, but It is not 100% perfect. The technique still has a lot of scope of development and improvement because there are several different categories of bacteria. If you ask what are the different types of bacteria according to the gram staining procedure to the students, they will generally answer that gram positive and gram negative. But there are also uh, two other classes, right? They are gram neutral and gram variable. So gram neutral and gram variable species are also there, but most of the students are not at all aware about them. So gram neutral spirochetes and gram variable means the bacteria that is displaying the characteristics of gram positive also, positive bacteria also, but also sometimes it displays gram negative organism also, right? So all these type of bacteria, they come under the uh, umbrella of actinobacteria or Gardnerella. Actinobacteria, which share the characteristics of both fungi and bacteria, are widely distributed in both terrestrial and aquatic ecosystem in many in soil, where they play an essential role in recycling refractory biomaterials by decomposing complex mixture of polymers in dead plants and animals and fungal material. They are considered as the biotechnologically valuable bacteria that are exploited for its secondary metabolite production. So, since biotechnology and microbiology evolved a lot, and when they discovered the potential of actinobacteria, they started working on it. And since then, the scientists were able to discover a lot of advantages. Most importantly, their secondary metabolite production. They are mainly employed in the production of medicines, right? So they have started to exploit more and more these actinobacterial species and trying to learn um, them properly about their behavior and about their nature. So approximately 10,000 bacteria, bioactive metabolites are produced by actinobacteria, which is about 45% of all bioactive microbial metabolites. So it is estimated that 10,000 bioactive metabolites are specially produced by actinobacteria, and which is about 45% of all bioactive microbial metabolites that have been discovered. So this significantly contributes towards the research aspect and the research scope of actinobacteria and in what direction things are going. Especially Streptomyces species produce industrially important microorganisms as they are a rich source of several useful bioactive natural products with potential applications. So, Streptomyces are also proven to be the types of industrially important microorganisms as they are serving to be a rich source of bioactive natural products with the potential application. So, this should be known that they do pack a heavy punch when it comes to the scope and research on medicinal compound and secondary metabolites. Though it has various applications, some actinobacteria have its own negative effects against plants, animals, and humans. So the majority is pretty has been proven to be pretty much useful for humans, but there are also some exceptional cases that have proven to be negative for the living organisms. In this context, the general characteristics of actinobacteria, its habitat, systemic classification, various biotechnological applications, and negative impact on plants and animals. They are also responsible for subsequent decomposition of humus. the resistant material in soil, and for earthy smell of freshly ploughed soil, and producing a number of antibiotics like streptomycin, tetramycin, uh, oreomycin, and so on. So, actinobacteria, especially streptomycin species, are recognized as the producer of many bioactive metabolites that are used for humans in medicine. They are very much useful for humans in medicine, such as actin, antibacterials, antifungals, antivirals, antithrombotics, immunomodifiers, anti-tumor drugs, and enzyme inhibitors, and in agriculture, including insecticides, herbicides, fungicides, and growth promoting substances for plants and animals. So they do produce a lot of compounds. They do have a wide range of applications, and many of them are proven to be antiviral and antifungal in nature. So they do have insecticide properties and anti-tumor properties, and many of them are enzyme inhibitors also. So growth promoting substances for plants and animals. So they do have a large spectrum of beneficial and harmful substances because they do create a lot of specifications and the genre of spectrum that can be helpful for human also, but also can be poisonous to human beings also. Actinobacteria derived antibiotics that are important in medicine include aminoglycosides, anthracyclines, chloroamphesinol, microlide, tetracycline, etc. Actinobacteria have high guanine and cytosine content in their DNA. The GC content of actinobacteria can be as high as 70%. Through um, some may have a low GC content, right? Analysis of glutamine synthetase sequence has been suggested for phylogenetic analysis of actinobacteria. So, what scientists are doing nowadays, they are just studying the genome and GC content of actinobacteria, right? What are the genes that are beneficial, or what are the genes that are producing these secondary metabolites that are extremely important to humans? So, 70%, they say some of them can be high, some of them can be low, and may have low GC content. Analysis of glutamine synthetase sequence has been suggested for phylogenetic analysis of actinobacteria. So, habitat of actinobacteria. Terrestrial environment. Soil remains the most important habitat for actinobacteria, with streptomyces existing as a major component in its population. According to numerous reports, Streptomyces was encountered to be the most abundant genus isolated in each study. So, Streptomyces is found in the majority of the experimentations when the samples were taken from the soil. Terrestrial actinobacteria have various interesting antimicrobial potential. Osca isolated actinobacteria that has had capability of producing novel antibiotics with high antibacterial activity. In anoxic mangrove rhizosphere, actinobacterial species such as Streptomyces, Micromonospora, and uh, Nocardioform were found to be abundant, which is 1,000 to 10,000 times smaller than arable lands because of tidal influence. Similarly, uh, Nocardia isolated from mangrove soil produce new cytotoxic metabolites that strongly inhibit human cell lines, such as gastric adenocarcinoma. So they do produce the components that are uh, anti-cancerous in nature, right? Desert soil is also considered as extreme terrestrial environment where only certain species, especially were in actinobacteria, often using micropolis as a source of food. There are several reports showing distribution of actinobacteria in various locations, such as sandy soil like Cairo, Egypt, and Falmouth, black alkaline soil of Karnataka and India, sandy loam soil in Metropolis, Kefi, Metropolis, Nigeria, alkaline desert soil in Wadi al Natron, Egypt, Wadi Arba in Egypt, and subtropical desert soil in Thar, Rajasthan. Where Streptomyces species were dominant, followed by other organisms such as um, Nocardia and Nocardiopsis and Actinomyces. Aquatic environment. Actinobacteria are widely distributed in aquatic environments, which may sometimes be washed in from surrounding terrestrial habitats. It is vitally important that the number of and kinds of actinobacteria are interpreted in the light of information on organisms such as uh, Anthero, Thermoactinomyces and Rhodococcus corpophilus, which are known to be good indicators of the terrestrial components of actinobacterial propagules in water and sediments. So these two varieties are known to be indicators, right? The resistant endospore of Thermatoactinomyces are produced in self-heating composts, overheated fodders, and surface soils. They can be washed into aquatic habitat where they are deposited in muds and sediments. So their main feature is to uh, decompose the sediments. It has been assumed that these thermophiles are unable to grow at ambient temperature in most aquatic environments. Similarly, uh, the rest focal stage of our Corpophilus passes into freshwater and marine habitats, where it can survive but probably does not grow. General characteristics of Actinobacteria. Actinobacteria comprises of a group of branching unicellular microorganisms, most of which are aerobic, forming mycelium known as substrata or aerial. They reproduce by binary fission or by producing spores of conidia, and sporulation of actinobacteria is through fragmentation and segmentation of or conidia formation. Right. The morphological appearance of actinobacteria is compact, often leathery, giving a conical appearance with a dry surface on culture media, and frequently covered with aerial mycelium. So here in this image, what you can see, the appearance of actinobacteria isolated on starch casein agar plate in first image. And image C, plate view of actinobacterial isolates. So these are the pure culture. And B and D indicates the morphology of individual colonies, how individual colonies are different from each other. Aerial mycelium. The aerial mycelium is usually thicker than the substrate mycelium. The aerial mycelium shows sufficient differentiation that the miscellaneous assortment of isolates can be segregated into a number of groups having similar morphological characteristics under fixed conditions. That is designated as one of the most important criteria of classification of genus Streptomyces into species, comprising structure, cottony, velvety, or powdery, formation of rings or concentric zones, and pigmentation. Substrate mycelium. The substrate mycelium of actinobacteria varies in size, shape, and thickness. Its color ranges from white or virtually colorless to yellow, brown, red, pink, orange, green, or black. Abundant growth of actinobacterial isolate on starch casing agar medium. So in A image, what they are showing that aerial mycelium. B, reverse side of plate showing substrate mycelium. So morphological appearance. Morphology has been an important characteristic to identify actinobacteria isolates, which was used in first descriptions of Streptomyces species. This is made using various standard culture media, including International Streptomyces Project (ISP) for non-Streptomyces or rare actinobacteria strains maintained on ATCC medium number 172 were used. Various morphological observations, including germination of spores, elongation and branching of vegetative mycelium, formation of aerial mycelium, color of aerial and substrate mycelium, and pigment production, have been used to identify actinobacteria. Light microscopy was used to study the formation of aerial mycelium and substrate mycelium, and scanning electron microscopy was used to study the spore surface, and spore structure. So here what you can see, type of spore bearing structure in Streptomyces. It can be straight, it can be flexus, uh, fascicled, monoverticulated, no spirals, open loops, primitive spiral hooks, open spirals, closed spirals, monoverticillate with spirals, biversillates, no spirals, and biversillate with spirals. So these are the different spore-bearing structures of Streptomyces. And in this image, what you can see is scanning electron photographs of various actinobacterial isolates. In A image, you can see uh, a micro monospora species, and in B image, you can see Streptosporangium species, and C image displays the Saccharopolyspora species, and D image represents Actino uh, Cinema species, and E image Streptomyces nosoria DPTD21, and F image displays Streptomyces species. So there is a lot of variation that you can see. Type of actinobacteria. Thermophilic actinobacteria. Number of studies has been carried out by the researchers to confirm the existence of extremophilic and extreme tolerant soil actinobacteria that are acid tolerant and alkali tolerant, uh, psychrotolerant and thermotolerant, and halotolerant and haloalkylotolerant, or xerophilic in nature. Mesophilic uh, actinobacteria can grow at an optimal temperature of 20 degrees to 42 degrees, among which thermotolerant species exist, which can survive at 50 degrees centigrade. Moderately thermophilic actinobacteria have an optimum growth at 45 to 55 degrees centigrade, whereas strictly thermophilic actinobacteria grows at 37 degree to 65 degree centigrade temperature, with the optimum temperature at 55 degrees centigrade. Incubation temperature of 28 degrees centigrade, 37 degrees centigrade, and 45 degrees centigrade are considered optimal for isolation of soil, mesophilic, thermotolerant, and moderately thermophilic actinobacteria. Thermoactinomyces, which is present excluded from the order of Actinomycetes, are described as thermophilic forms depending on its phenotype and molecular genetic characteristics, as well as among some species of Thermomonospora, Microbispora, and Sacropolyspora, Sacromonospora, and Streptomyces. Acidophilic actinobacteria. Acidophilic actinobacteria, which are common in terrestrial habitats, such as acid forest and mine drainage soil, um, grow in the pH range from about 3.5 to 6.5, with optimum rates at pH 4.5 to 5.5. It has been shown that acidophilic actinobacteria consistently form two distinct aggregates taxa, namely the neutral-tolerant acidophilic and strictly acidophilic clusters group that are based on numerical uh, phenetic data. Members of two groups share common morphological and chemotaxonomic properties. Some members of strictly acidophilic group form a distinct taxon, such as the genus Streptoacidophilus, which can be assigned to the revised family Streptomycetaceae, together with genre Kitaspora and Streptomyces. Halophilic actinobacteria. Halophilic actinobacteria are characterized into different types based on their growth in media containing different concentration of salts. Extreme halophiles grow best in media containing 2.5 to 5.2 molar salts, whereas borderline extremohalophiles grow best in media containing 1.5 to 4.0 molar salts. Moderate halophiles grow best in media containing 0.5 to 2.5 molar salt, and finally, halotolerant that do not show an absolute requirement to salt for growth, but grow well up to often very high salt concentration and tolerate 100 gram per liter salt at least. Seawater, saline soils, salt lakes, brines, and alkaline saline habitats are considered best as the best habitats for isolating halophilic actinobacteria. Generally, most of halophilic actinobacteria have been isolated from saline soils. Halophilic actinobacteria isolate from marine environments and assigned to a few genera, including Micromonospora, Rhodococcus, and Streptomyces. Endophytic actinobacteria. Actinobacteria are defined as those that inhibit the internal parts of the plant, causing apparently no visible changes to their host. These actinobacteria play significant specific roles, uh, for instance, protecting the host plant against insects and diseases. Endophytic actinobacteria constitute a large part of rhizosphere, which are also found inside plants in which The extensively studied species are from genus Frankia, nitrogen-fixing bacteria, and non-leguminous plants, and few species of the genus Streptomyces are phytopathogens. Symbiotic actinobacteria: about 15% of the world's nitrogen is fixed naturally by symbiotic relationships between various species of Frankia belonging to the family of Actinobacteria. The plants that form symbiotic relationships with Frankia are called actinonoizal plants. Researchers have found that over 160 plants have Actinobacteria as their host, including alders, Russian olive, bayberry, sweet fern, brush, and cliff rose. The Frankia have the ability to provide most or all of the host plant's nitrogen needs. They help in nitrogen fixation. Numerous Frankia species, including uh, Fusariana, Ariana, isolates from nitrogen, including NIR, vesicles in vitro and in planta. These nitrogen-fixing bacteria and their host plants are often pioneer species on young nitrogen-deficient and distributed soils such as moraines, volcanic floors, and sand dunes. Here you can see the actinobacteria, and that is being used to produce different types of antibiotics like macrolides, glycopeptides, glycopeptides, polyene nucleosides, tetracycline aminoglycosides, chloroamphesinol, anthracyclines, beta-lactams, polyesters, polycatides, and peptides. Applications of actinobacteria. Actinobacteria are well recognized for their production of primary and secondary metabolites that have important application in various fields. They are also a promising source of wide range of important enzymes which are produced on an industrial scale. A large fraction of antibiotics in the market is obtained from actinobacteria. They produce enzyme inhibitors used for cancer treatment and immunomodifiers that enhance immune response. They have the ability to degrade a wide range of hydrocarbons, pesticides, and aliphatic and aromatic compounds. They perform microbial transformation of organic compounds, a field of great commercial value. Members of many genera of actinobacteria can be potentially used in bioconversion and underutilized agriculture and urban waste into high-value chemical products. Actinobacteria are also important in plant biotechnology as strains with antagonist activity against plant pathogens are useful in biocontrol. Their metabolic potential offers a strong area for research. Here we have a brief description about important applications of actinobacteria. So the conclusion: Actinobacteria is one of the dominant group of microorganisms that produce industrially important secondary metabolites. A wide range of antibiotics in the market is obtained from Actinobacteria. Products such as enzymes, herbicides, vitamins, pigments, larvicides, phytohormones, uh, surfactants are produced by these several genera of Actinobacteria, which are of great commercial value. They are capable of degrading a wide range of hydrocarbons, pesticides, and feather waste, and their metabolic potential offers a strong area for research. However, many of the rare genera of Actinobacteria have been neither discovered from unexplored locations nor employed for their biotechnological and industrial potential. Thus, studies on unique ecological environments could yield molecules that could become further harbingers of green technology. So coming on to the MCQs, lactobacilli grow best under microaerophilic conditions in presence of 5% CO2 and pH 6. Which of the following genera is included in the family of bacteria? They see all of these: bacterioids, fusobacterium, and leptotrachea. The bacteria which performs a predominant flora of human gut is Clostridium perfringens or E. coli. So, colonies resembling molar teeth are produced by Actinomycetes israeli. Current genus name of saccharolytic group of bacterioids are Prophyromonas. Which of the following bacterial colonies are fluorescent brick red light in UV light? B. Meninogenicus. Which of the following bacteria is acid fast? Nocardia. Lactobacilli constitute the normal flora of adult vagina. Which of the following bacteria is gram-negative foci? Uh, Bellinolia. Which of the common species of bacterioids in Fragilis group causes human infection in B. fragilis? These are the references from where we have collected the material. And thank you very much for listening to my lecture.